Okay.
J'avais présenté donc travaux de Luka et CULIG, donc travaux qui font l'objet qui sont l'objet et nous travaux, le professeur Pierre Vincent, le docteur Pierre Vincent, qui va à ma gauche, et le docteur David, qui se trouve à ma droite. Donc, vous aurez dans une quarantaine de minutes présent.
So first of all, thank you for being here. It's nice to see all the familiar faces. So let's just begin with the presentation. So the title of my talk will be The Effects of Increasing Adult Hippocampal Neurogenesis in a Naturalistic Mouse Model of Depression. And my presentation is structured, so I will go from the last part front. So first of all, we should distinguish between depression and sadness, because in popular culture, those two are often used interchangeably. Okay. Okay. They're used interchangeably. And the fact is, sadness is a transient feeling, one of the basic emotions related to resignation, and often occurs after threat or perceived loss of threat. While depression is a mood disorder classified by the DSM as having several symptoms. One of these two must be either a depressed mood or loss of interest or pleasure activities, also known as anhedonia. Also has some associated symptoms such as significant loss, weight gain or weight loss, suicidal ideation, and so on. The burden of depression on society is very high. It accounts to 7.5% of years lived with disability in 2017, and up to 2/3 of people who commit suicide suffer from depression, with rates much higher in women than compared to men. There are several treatments for depression, with psychotherapy being a common one. Usually it's cognitive behavioral therapy is used most often. Then we have pharmacotherapy with selective serotonin reuptake inhibitors being the primary choice. Then we have electroconvulsive therapy, which is quite effective, especially in treatment-resistant depression, but is not used often. And we also have some new methods of treating depression, such as repetitive transcranial magnetic stimulation and sleep deprivation. Notably, in the STAR-D study, it has been found that after the first line of treatment, which was an antidepressant, only 1/3 of people remitted from depression, while after the second level of treatment, which was an addition of cognitive behavioral therapy or a replacement of an antidepressant to another one, only one half of the population remitted. So this means there are valuable new treatment options which could be explored to better the treatment of depression. There are several alterations in depression, so all are affected. Most notably, these are the hippocampus, nucleus accumbens, which is often enlarged in patients with major depression, but these studies are inconclusive. And the medial prefrontal cortex has a lower activation compared to controls. So there are several hypotheses proposed to explain the psychological, cognitive, and behavioral alterations as a result of depression. The first and the oldest hypothesis is the Monoamine Hypothesis, which claims that an absence or a deficiency in the monoamine neurotransmitters is the culprit for depression. So this is usually related to serotonin and norepinephrine. Then we have the dysfunction of the HPA axis as one of the proposed mechanisms of the etiology of MDD. So it claims that a blunted negative the HPA axis is the cause of depression. So the HPA axis I will go into detail further, but it consists of the hypothalamus, the pituitary, and the adrenal gland. And the adrenal gland releases cortisol, which does a negative feedback loop on the HPA axis, which is blunted in a subpopulation of depressed patients. This theory says that inflammation responses might be the culprit for depression. The circadian rhythm disturbances are based on the fact that some patients who suffer from depression have a different circadian rhythm compared to the physical reality, in the sense that they're misaligned. And lastly, we have the neurogenesis hypothesis, which I will go into detail a bit later, but it claims that the birth of newborn neurons in the hippocampus might have to do something with depression. So just to return to the HPA axis shortly, basically the hypothalamus the hypothalamus is the one that releases the CRF after exposure to stress. In turn, stimulates the pituitary gland, which releases the adenocorticotropic hormone, which stimulates the adrenal gland, which then releases cortisol into the bloodstream. And cortisol is a stress hormone, which usually has a flight or fight response. And the cortisol binds to glucocorticoid and mineralocorticoid receptors, which serve to blunt the feedback. So after the cortisol has been released, it negatively affects almost all of the aspects with two mechanisms. And one of the areas which is important in this feedback is also the bed nucleus of the stria terminalis, which regulates of the hypothalamus, coming back to this loop, which I described right now. So regarding the neurogenesis hypothesis of depression, it consists out of two levels. One claim is that reduced neurogenesis leads to depression and anxiety. Experimental evidence do not really confirm this part of the hypothesis. And the second level is that antidepressants act through these newborn neurons and increase their number. So there are several points to the mechanisms in depression. We found that neurogenesis, while in contrast, antidepressants increase it. One study has established a causal link, which has shown that for the effects of SSRIs, newborn neurons are activated. And this view has later been, it's more subtle because there are also some neurogenesis dependent and some neurogenesis independent effects of antidepressants. Lastly, chronic cortical model, newborn neurons that increasing neurogenesis is enough to remit some of the animals who have been exposed to stress. So regarding animal models of depression, I will just shortly mention the most commonly used ones. So these are the learned helplessness model, which consists of placing the model in an arena and administering inescapable food shocks. After a while, an animal develops resignation behavior and depressive-like behavior. We have maternal separation, which consists of, it's an early life intervention where pups are separated from their mothers, and during adulthood, they usually develop depressive-like symptoms as well. We have social defeat stress, which has many different, but the most commonly one used is the resident intruder paradigm, in which an animal is introduced to a resident of a cage, which is usually larger, bigger, more aggressive, and after repeated exposures where the animal is defeated, the animal which was introduced develops submissive-like behavior, which is related to depressive-like behavior. And we have the exogenous corticosterone administration, which is based upon the fact that the HPA axis negative feedback is usually blunted in a subpopulation of depressed patients. And lastly, we have the unpredictable chronic mild stress. This is the model which I used in my experiments, which consists of administering unpredictable and mild stress for some time, which mimics well the naturalistic progression of depression. So, yes. Finally, the objectives of my thesis. So, just one sentence. I wanted to investigate depressive-like behavior, basal and not just basal, but yeah, corticosteroid levels, activation after increasing adult neurogenesis. So basically, a lot of things have been shown in the neurogenesis field, but it hasn't been established if just increasing adult hippocampal neurogenesis would be enough to revert the depressed mouse to a baseline behavior. So we have two main questions. So is increasing adult hippocampal neurogenesis after exposure to stress sufficient to protect against the effects of stress, which is just differently worded statement, and elicit their effects. To tackle these questions, and these mice enable us, which is important for apoptosis. Essentially, what we do with this model is, after administration of tamoxifen, we block apoptosis in neural stem cells because it's bound to the nestin plus promoter. So only nestin cells, aka neural stem cells. The timeline of the experiment was as described. So it's a 10-week-long UCMS protocol. In the third week, we exposed the animals. We administered tamoxifen or vehicle. We waited for 4 weeks before behavioral testing, after which we have done surgery. By surgery, I mean intrahippocampal cannulas, because we wanted to do the dexamethasone suppression test. So the dexamethasone suppression test is a test enabling us to probe the HPA axis because the dexamethasone is a glucocorticoid receptor agonist. So after it's bound to the glucocorticoid receptor, testosterone levels should be lower, and if they are not, this means that the HPA axis is not functioning properly. So, and we wanted to do intrahippocampal dexamethasone suppression test because we wanted to distinguish the local from the global effects, because we wanted only to target the glucocorticoid receptors. And in week 10, the animals were perfused. So these are the results. We used DCX, or double cortin, a marker of immature neurons, to establish the differences in neurogenesis. Basically, what is that stress was able to decrease the levels, while tamoxifen was able to revert this level even after exposure to stress. Regarding the behavioral effects, we found that stress significantly worsened the code state of animals. So these two lines are actually stressed animals, while these are the non-stressed animals. They all progress towards worse code state, but there's a significant difference between stress and non-stress, which means the stress protocol worked, at least in part, regarding to the code state. Interestingly, in week 9, we see this point, which is the UCMS tamoxifen point, which is significantly different from the UCMS vehicle point. A lower score at the code state means less degradation of the code state. So this means in week 9, tamoxifen or increased neurogenesis was able to revert the effects of stress, partially. Then we checked for locomotor activity and found that there are no differences between group, which means all the other tests, the results from all the other tests are not a consequence of a change in locomotion. In the nest building test, it's a test which assesses spontaneous motivation. We found a kind of paradoxical effect where stress was able to increase the scores of nest building. We usually in the UCMS protocol see that UCMS decreases the scores of nest building, and these animals are slower to build well-built nests. But in this case, we found an opposite effect. And the interpretation is that it was an acute stressor for non-stress mice to transfer them from group housed to a new cage, which was essentially empty just with a nestlet, while it was an improvement in conditions for UCMS-treated mice, thus enabling them to quickly establish a well-built nest. In the light dark box, where we establish anxiety-related behavior, we find the same pattern with stress decreasing time spent in the dark box. So time spent in the dark box is related to anxiety-related behavior, and the longer the animal, the longer it is in the dark box, it means it's more anxious-like behavior. So stress was able to decrease this behavior in both vehicle-treated groups and tamoxifen-treated groups. So this is also similar to the nest building test, and we explain it in the same way as an acute stressor for non-stressed mice, while it's improvement in conditions for stressed mice. And interestingly, we found that there's a difference between UCMS vehicle and UCMS tamoxifen. So tamoxifen even further decreasing the time spent in the dark box, meaning these mice display less anxiety-related behavior. Then we did the splash test. It's a test which also measures motivation. And the variable we looked at was the duration of grooming. And we found that UCMS was able to induce an increase in the duration of grooming. So this is something which is not often found in UCMS protocols. In animals exposed to UCMS, usually UCMS decreases the duration of grooming, but also some researchers found that the duration of grooming can be increased after exposure to stress. In our case, we proposed that the animal could have overgeneralized the splash of sucrose on its coat and wasn't able to disengage the behavior in time, thus increasing the duration of grooming. And we don't see this difference in tamoxifen-treated mice, which we speculate could be related to tamoxifen lessening the anxiety of these mice. Lastly, we did the sucrose preference test to establish if there is a difference in anhedonia, but we failed to see any difference between groups. For endocrine effects, if there is a blunted negative feedback in the HPA axis, since in the normal conditions, so non-stress vehicle conditions, we found, so basically what we compared was the levels of corticosterone after saline administration, and we found a difference between UCMS vehicle and UCMS tamoxifen, with tamoxifen significantly decreasing the levels of basal cortico, which means that in vehicle groups, stress was in UCMS tamoxifen-treated mice. Regarding the mechanism of this change, we checked substrate. The only difference we found was that there is a lack of treated mice after dexamethasone stress. So in all pairwise comparisons, there is always a decrease after stress. So this one, and this one, while this change is absent after exposure to stress and dexamethasone in UCMS condition. So this could be a potential pathway through which the HPA axis was modulated, because the hippocampus itself projects to the bed nucleus of stria terminalis, so newborn neurons could have affected it, and by that the HPA axis as well. So what are the conclusions of this study? We found that by increasing adult hippocampal neurogenesis, we were able to counter, but not all deficits. We were able to protect against stress-induced elevation in basal corticosterone levels, and we found changes in neural activation in bed nucleus of stria terminalis, which are not as straightforward, which then leads us to study 2, where we wanted to explore in detail how do newborn neurons elicit their effects brain-wide. We had a similar protocol to the first one. It's just 1 week shorter because we didn't do surgery in this experiment because we wanted to have the brains intact for whole brain image analysis. Also added wild-type mice, so tamoxifen will not be able to excise backs from these mice and thus doesn't increase neurogenesis. Again, in week 3, we administered tamoxifen or vehicle to these animals, waited for 4 weeks to do behavioral tests, with cookie tests being a bit before. And in week 9, the animals were sacrificed. So regarding adult neurogenesis, again, we used the exact same protocol as in the study 1, but we found no significant changes in double cortical. We tried to do age of the animals. So there were some animals in study 2, the young ones and the old ones. The young ones were 13 weeks at behavior, and the old ones were 18 to 22. And this difference will establish a very big difference in neurogenesis. So, while in the young ones, the neurogenesis is quite high across the board, we essentially replicate the findings we found from the first study. And tamoxifen in stress condition was able to reverse this effect. What we propose, levels of neurogenesis in young mice were already so high, treatment was unable to increase them further from this plateau, because the levels here are quite high compared to the older population of mice. Regarding the behavioral effects, we found that stress was able again to worsen the cognition of these animals compared to non-stressed animals. But in week 9, we weren't able to distinguish between UCMS tamoxifen and UCMS vehicle. But again, this is to be expected because we found no differences in neurogenesis. Yes, I should mention that all the behavioral effects and everything I will be talking after this is all groups. So we didn't distinguish between young and all animals. And this was necessary because of the later connectomic analysis, which would suffer from a too low sample to do any meaningful or significant statistical correlations. So these are all animals included. In locomotor activity, we found no differences in locomotion, which means again that the results from the shouldn't be a consequence of basal changes in locomotion. In the nest building test, we replicated the findings from the first study with UCMS exposure to stress increasing the code state again. And in the tail suspension test, which is a measure of behavioral despair, interestingly, we found that tamoxifen in non-stress groups was able to decrease the immobility time between minutes 4 and 6. This is difficult since tamoxifen didn't elicit its effects on neurogenesis, so it could have been a non-specific effect. We did the cookie test across 5 sessions. So this is a test which measures anhedonia-related behavior. We failed to find any significant statistical changes. Everything we found was just in week 3 between UCMS vehicle and UCMS tamoxifen, so these 2 points. And this is the number of bites. So the number of bites was higher in UCMS tamoxifen, and likewise the number of smellings of the food. But the p-value wasn't as close to statistical significance. It was 0.075, more or less. So any asterisks here. It's just kind of a trend of tamoxifen was able to produce some kind of an effect in anhedonia-related behavior. And lastly, we did the light dark box test again. So this is time in dark box, again a measure of anxiety, where we found there is a trend between non-stress vehicle and UCMS vehicle mice, with stress decreasing the amount of time spent in the dark box. So this is as this is a measure of anxiety-related behavior, it's also quite difficult to apprehend as a kind of a paradoxical effect, because stress should increase the time usually in the dark box, but in our case we found a trend toward a decrease, which again could be maybe explained by being an acute stressor for non-stress mice, while an improvement of conditions for stress. For endocrine effects, we checked only basal corticosterone, so we didn't do dexamethasone suppression tests. And we found a surprising effect that stress was able to actually decrease the levels of basal corticosterone after exposure of tamoxifen, both in and heterozygous mice. First of all, this usually stress would increase the levels of corticosterone, but this could be due to the fact that non-stress mice were handled much rarely compared to the UCMS mice. So they were handled once, twice per week in assessing code state and changing their food and bedding, while UCMS treated mice were handled multiple times daily. So when it time came to assess their corticosterone levels, maybe the non-stressed mice were much more stressed because of they weren't so used to the handling, while for UCMS-treated mice, UCMS-exposed mice, it wasn't such a big deal, hence lowering the levels of basal cortisone. However, it occurs only in tamoxifen mice, both in heterozygous and wild, which again could point to a non-specific effect of tamoxifen. So in the first study, we studied the bed nucleus of the stria terminalis to check if the HPA axis is regulated through it. In this case, we wanted to check the whole brain or as much as possible. Using CFOS, like in the previous study, we used delta FOS-B. So it's a neuronal marker for chronic activation of neurons, especially after stress. So it's a splice variant of FOS-B, a transcription factor. And unlike CFOS, which has a steep induction rate and very quickly goes to baseline, delta FOS-B stays in the cell for a prolonged time. And most importantly, after exposures to stressors, it accumulates in the cell. So it's a very good marker to study stress-based chronic activation. To count the cells, it's quite difficult here to see. This is one section with inverted colors with all the cells. Here, there should be computer-counted cells, and these are the regions of interest which were where the counting was performed. So we took the number of cells detected via a custom script, divided by the area, and used cell density as a measure of activation. We used the graph theoretical approaches to find differences between different groups. So first of all, to explain just the difference between the node and the connection. Node is any brain, such as the amygdala, nucleus accumbens, and so on, while a connection is its statistical correlation to another area. So the correlation between activation of nucleus accumbens and amygdala, for example. There are many measures which can be used in this graph theory, but we focused on two of them, one of them being degree. So degree centrality, if we look at this blue node, it has a degree centrality of 5, which means it projects to 5 nodes, adjacent nodes, I should say. And we also have betweenness. Betweenness centrality is a measure of how many shortest paths go through this. So in this case, under this 4 would have very high betweenness because it would be very difficult from the one end of the network to come to the other end of the network. So they would have very high betweenness. Maybe this example is even better because this one would have extremely high betweenness, as it's not possible to come from any of these nodes to these nodes. So it's just a number of average shortest lengths through this node. So we did correlation matrices for each of the groups, animals per group. Initially, we wanted to check 80 brain areas, but we subset them to 66 areas, which were present in all animals. And basically, the color coding here is the more red, the more the warm color is, the higher the correlation statistical one between two areas is. So each is on each axis. What else should I mention? Yes, and the blue colors are negative two areas. And what we see here is that areas which are here around number 30, so these are the hippocampal areas and the lateral entorhinal cortex. They have negative correlations with the nucleus accumbens, and this has not been found in any of the other groups. So it's interesting that stress was able to induce a negative correlation between those two areas, especially since nucleus accumbens is implicated in reward-related behavior. So after we have done these correlation matrices, we thresholded them to only keep the most significant connections. We only kept the top 10% of all connections, and we generated graphs from these. So each line here represents a correlation between activations of these two areas. We divided all the areas into 5 different groups. So we have cerebral nuclei, the hippocampus, the thalamus, hypothalamus, and the neocortex. And this is just a visual inspection. Groups, everything more or less connected. And I should also say the size of the node corresponds to the degree of the node. So the bigger the is, it is to other nodes, and the color corresponds to the community it belongs to. So this was only done to check if our functional connectome might be somehow connected to physical reality, because if it was, then a lot of the same colors should be under the same dashed lines. Okay, so we failed to see anything very interesting here. Everything is more or less connected. But after exposure to stress, we find that the thalamus is engaged much more compared to here. And while the neocortex kind of disengages and doesn't have as many connections as it does in the non-stress vehicle conditions. We find something similar in non-stress tamoxifen-treated mice. Essentially, everything is pretty well connected with nothing standing out. But again, after exposure to stress, we find this informational highway between the cerebral nuclei and the thalamus, while the neocortex is almost completely disengaged from the network. So we ordered all the nodes, all the 66 of them by degree, and we only kept the top 90th percentile. So these were the nodes which we determined that have the degree interesting for us. We did the same for betweenness, and Venn diagram an intersection of those two to find the most important regions of the brain, like information hubs. So this is just done for non-stress vehicle, UCMS vehicle, non-stress tamoxifen, and UCMS tamoxifen. When we look at top nodes by degree, by group, we find that in non-stress vehicle group, so everything which is orange is the neocortex. We find a very strong picture and a bit of the hippocampus. This corresponds well also to non-stress conditions in tamoxifen-treated mice, also neocortex and hippocampus. After stress changes dramatically, neocortex isn't so much involved anymore. Hippocampus is engaged. When we also have an engagement of the thalamus, especially interestingly the shell and the core of the nucleus accumbens. After tamoxifen treatment, again we find a completely different induction pattern with thalamus, hypothalamus, and cerebral nuclei being active, and a complete disengagement of the neocortex. We've done the same for betweenness. Here we find not so dramatic changes, with neocortex being equally important in all of the groups, similarly with the hippocampus. But we see as stress progresses, for example, in UCMS tamoxifen-treated groups, the cerebral nuclei get more activated, and we also have some engagement of the hypothalamus. Yes, so betweenness is not as much as informative as degree. However, when we did hub identification to check for the information hubs, which were, by the theory, most important in information processing of the network, so a Venn diagram of both degree and betweenness, we found that most of the nodes were actually quite different amongst groups. In non-stress vehicle, again, the most important ones are neocortical and hippocampal ones. Similarly, in this non-stress group with tamoxifen, so neocortex, hippocampus. After exposure to stress, we found a thalamic signature and also a cerebral nuclei. Interestingly, after exposure to tamoxifen and stress, there is only one node which is important in this group. So this is the zona incerta, which hasn't been really explored so much in depression. It's usually related to Parkinson's disease and central pain. If this analysis is valuable, it could be an interesting area to focus on in later studies. So the conclusions of the second study is that we failed to induce an increase in adult hippocampal neurogenesis across animals. We were able to do it in the older population of mice, but not in the younger one. We reported paradoxical findings related to behavior, but also to endocrine levels, so stress actually decreasing the levels of basal corticosterone. We found there are different active hubs across different groups, and that stress induced a strong thalamic signature. Okay, so the perspectives of this work. So this is especially related to the second study. The first point, we could have better characterized newborn neurons. So we could have administered BRDU, and we could have checked for tertiary dendrites to establish the cell maturation of these newborn cells, because it's possible that the maturation is also different, and it would be nice to see how functionally integrated these newborn neurons are as a result of the be good also to compare the results to an antidepressant. So I proposed two of them because one of them has neurogenesis dependent and one of them has neurogenesis independent mechanism effect, so fluoxetine and almorexant. This would enable us to distinguish better which behavior is more neurogenesis based and not, and also to establish the strength of the newborn neurons in relation to behavior, so the effect size, and essentially. We could have better characterized the active neurons in the bed nucleus of stria terminalis, or the paraventricular nucleus. So this is related to the first study. We could have used GAD65, which is only present in cells which synthesize GABA, so an inhibitor, as it's not really the same if the same number of neurons is activated in bed nucleus of stria terminalis, but some of them are excitatory. And good to know the exact identity of these cells. So we could have minimized exposure to an acute stress cohort levels, essentially handling non-stressed mice daily as much as UCMS-treated mice are handled, could have decreased this acute stress of blood taking, or we could have done a fecal collection of corticosterone, which would be much less effective on the animals. It would be good to optimize the length and the severity of the UCMS protocol because it seems that ibex mice are quite stress resistant, and we observe the effects on code state only in week 9, which might mean that this is when newborn neurons functionally integrated in the network, and maybe they would elicit stronger effects as time went by, and just increase the frequency of mild stressors, so M, in an attempt to stress them even further. So what should be done is to eliminate variability in age of animals. We didn't know prior to taking this study that in young animals we wouldn't have this effect. And it would also be good to check for optimal levels of adult neurogenesis, because it could be that supraphysiological levels might be actually bad for the animal. And since newborn neurons have have effects on pattern separation, it's always a battle between plasticity